Good morning. Thank you so much for the kind introduction. And I just want to say very briefly, thanks to the organizers for this wonderful opportunity. So after decades of development, today's anti-cancer cellular immunotherapies are now breakthrough modalities in the treatment of cancer, but still are in early days and are currently limited by ballistic dynamics. Today's CAR T cells, for example, after a fusion, can proliferate extraordinarily rapidly and reach an early peak at which many billions of cells autonomously, simultaneously kill and inflame and then contract variably afterwards. With such limited personalization, unfortunately, a subset of patients suffer very severe toxicities, as in red, whereas numerically a larger subset of patients fail to benefit because of undertreatment in gray. And as we adapt these modalities for targeting of solid tumors and more complex clinical scenarios, it's becoming increasingly clear that simply encoding a chimeric antigen receptor, an engineered TCR for tumor targeting, is not going to be sufficient. And what will be required to achieve durable clinical responses will be the incorporation of additional programs that enhance the potency and the persistence of these cells. So to summarize all of that, one of the overarching challenges to this field then is to learn how to simultaneously both enhance the safety and the potency of the next generation of cell therapy designs. We're preparing to program cell therapies with excess potency as well as precise control so that they can be tuned down or up from day to day as clinical scenarios change. And to do so, new toolkits are going to be required in order to control a cell's effector functions, growth, differentiation, and additional factors. And ideally, these programs would be encoded with molecular switches, molecular circuits, that are all of immediately acting, reversible, composed of human sequences, and controllable with approved drugs that are either close to inert or even beneficial in the clinical scenario. And so where can we look in the human proteome and the human genome for the building blocks for this next generation of technologies? And this biology was already very elegantly introduced by Dr. Haggerty, so very briefly, I think that our human protein degradation system very uniquely satisfies these criteria. And so here I'm showing you an E3 ligase complex that acts to mark targeted proteins, like the one in light blue, with ubiquitin for subsequent proteasomal degradation. And these are exceptionally modular complexes that really are akin to power tools in that they have a conserved catalytic core, and then many interchangeable terminal elements in green, like so many drill bits that can specify the precise functions and targets of these complexes. Really a fundamental biochemical language that we are increasingly able to control with small molecule drugs. So in this example, just visible intercalated at the surface between the blue and the green is a thalidomide analog that acts as a molecular glue to recruit a new set of substrate proteins whose degradation really underlies the mechanism of action and the efficacy of these drugs. Thalidomide, lenalidomide, pomalidomide, this class includes frontline, FDA approved, in general, with important caveats, well tolerated drugs that are frontline in the care of hematologic malignancies, a number of hematologic malignancies. And so these ternary complexes, these ternary protein-drug protein interactions are the starting point for our engineering efforts with which we create new synthetic systems, inducible degradation, inducible proximity, and other systems that then allow us to encode smart precision cell therapies. So next, I'll take you through 3 short vignettes. First, we engineered a very small, a 60 amino acid, all-human, zinc finger-based Degron tag that can be affixed to different engineered proteins. And in this example is connected, fused in frame to an otherwise conventional chimeric antigen receptor. And so this very minor modification to this modality allows for very efficient off-switch control. So these receptors function normally in the absence of lenalidomide, and then upon addition of the controller drug, are very robustly recruited to the E3 system for ultimate degradation. And so what I'm showing you on the right is that there is very efficient degradation of these CARs in the nanomolar drug range, which is 2 orders of magnitude below the concentrations required to degrade key endogenous substrates like IKZF3, Icaros, and 3 orders of magnitude below the concentrations that are maximally seen in patients with initial myeloma dosing, for example. And so this, in data that I won't show you, this system has allowed us in preclinical mouse models to pause the activity of these chimeric antigen receptors at the peak of their cytokine release, and then allow them to reactivate upon washout of the drug several days later and complete their anti-tumor function. Because these drugs are very effective anti-tumor agents, also with T-cell immunomodulatory factors that could be beneficial in these settings, we built an on-switch system to be able to combine the orthogonal cell and drug-based activities. And so we extracted and then further engineered the minimal protein domains that interact with these drugs, in this case pomalidomide, but interchangeably with the other small molecules, and then encoded these elements within a splitcar architecture in which the antigen binding domain and the signaling domains are on separate polypeptides. So only in the presence of drug are these split receptors approximated and licensed to activate in the context of antigen. And in a first in vivo mouse model demonstration, first in the center panel with the control T cells, you can see essentially the unopposed expansion of the luciferase positive tumor cells with or without pomalidomide treatment, whereas on the right-hand side with the splitcar, you can see the drug-dependent anti-tumor effect. We've also moved on to tuning additional aspects of T-cell biology that are important to the future of these cell therapies, their proliferation, cell state, response to tumor microenvironment, to both explore specific therapeutic opportunities and also demonstrate the generalizability of our systems. And so in this case, we've explored a number of different cytokines that can be tethered to the cell surface and rendered drug controllable with transmembrane and Degradation domains. And this, again, allows for small molecule control of the stability and also the signaling of these cytokines at sub-therapeutic drug concentrations. And so with the first example of IL-7 that allows us to independently control the proliferation as well as specifically enhance the naive memory, less differentiated subsets of these cells, this transgene allows us to quite dramatically enhance the anti-tumor activity of this system. And so in this mouse model, again, I'm showing you first in black the unopposed very rapid expansion of luciferase positive tumor cells when only treated with untransduced T cells, the delayed dynamics of tumor expansion when treated with a low subcurative dose of conventional CAR T cells, and then in this case long-term anti-tumor control with the addition of the controllable IL-7 element. And so for this is a first demonstration that we can now enhance the potency of these cell therapy systems while also attending to the need for greater safety and personalization that I think has been a little bit lagging in the development of these modalities. So this platform is ready for clinical testing, certainly a little bit earlier than some of the very developed new companies that you've seen in previous presentations. And so we're at the point of assembling these multiple genetic programs that all have been developed within MGH and MGB. And now as an ensemble, we can de-risk novel and highly active elements, novel and highly active combinations with this additional layer of pharmacologic control that is very amenable to incorporation and engineering within existing vector designs. And so leveraging the translational expertise and infrastructure of the MGH cellular immunotherapy program, we're seeking investment to complete first-in-human phase 1 trials at MGH with the option of licensing to advanced promising candidates. And so to summarize, immune effector cell therapies are a transformative, incredibly exciting, emerging pillar of cancer medicine, and also to date, of course, the smallest of these pillars. And so to broaden their scope, additional synthetic biology technologies are going to be needed. And targeted protein degradation, I think, is and really will continue to be a major resource, not around a single small molecule family, but really a class-wide endeavor as the drug discovery field of targeted protein degradation continues to expand incredibly rapidly. And finally, with these tools and with additional programs to target tumor antigens and control the biology of immune cell programs, we're seeking partners to advance first-in-human clinical trials here at MGH and deliver these advances for the treatment of people with cancer and limited treatment options here at MGH. Thanks so much.